Hi everyone. I'm a joint PhD student with the University of Adelaide in Australia and the University of Nottingham in the UK. I spent the last 2 years in Adelaide, and today I'm going to present some of the work that I did there. Basically, I wanted to look at frequency versus quantity. So how do roots adapt to variability of both water and nitrogen stress? So we know that the current climate is changing. It's changing faster than plants' ability to adapt. Therefore, genotypic variation is becoming increasingly important, especially in crop species, if we're to try and increase and improve food sustainability. One genotypic variation, this can be seen as plant plasticity. So this is basically the plant's way of adapting to its environment. And this is affected by many different things, but for the purpose of this talk, I'm just going to focus on two things, which would be frequency and quantity of resources. So we know that water variability directly affects nitrogen availability. So in essence, water in the soil is going to affect how the nitrogen cycle works, and so is going to affect the conversion of your nitrogen fertilizers into forms that plants can uptake, namely ammonium and nitrate. And because of the current climate, rainfall is becoming more erratic. This can be seen in a variety of different ways, but one way which I'm sort of looking at is high rainfall over a short period of time, followed by no or very little rainfall over longer periods of time. Therefore, we need to try and understand how the timing of rainfall or irrigation or supplementary irrigation could then affect crop nitrogen supply and uptake and ultimately crop yields. So while I was in Adelaide, I was really lucky to be able to use this unique system. So this is called the drought spotter. So essentially what it does is it precisely maintains and measures pot weight in real time, and then it irrigates each pot to a pre-programmed water level. Pretty nifty. So basically what happens is that each pot sits on its own weighing scale, and it's got a tap attached to it. And then what it does, the computer then sort of weighs it the computer then tells the scale to weigh every 10 minutes or so, and then water the pots accordingly if the pot reaches a weight percentage below the threshold target weight. So I decided to have a look at two Australian spring wheat varieties, Gladius and Kukri. So Gladius in the literature has been shown to be drought tolerant and was actually one of the best performing varieties in Australia, some of Australia's worst droughts in the 2000s. And Kukri, on the other hand, is more of a drought susceptible variety and also been shown to be more nitrogen use efficient as well. So I grew these two varieties in 2.5 kilo pots, and I used a semi-artificial soil medium. So this was a 1:1:1 ratio of cocoa peat, clay loam, and UC sand mix. And this was homogeneously mixed and then potted up. So as I said before, the unique thing with the drought spotter is that I could set up as many water treatments as I wanted. So what I did is I set up 5 water treatments. So basically, all my soils were pretty wet at the beginning, so they all started wetter than I wanted them to. So then I dried them down until they hit my baseline wet treatment. So this is where the soil was at 100% of field capacity. Now, for those of you who don't know what field capacity is, essentially it means that all the soil pores in the soil are filled with water, but no water is leaking out of the soil volume. In addition to my wet water treatment, I also had dry water treatment. So this is where some of the soils were dried down until they hit 50% of field capacity and then maintained at that level. And then I also had 2 cyclic watering treatments, one at wet, so these were watered every 48 hours as opposed to every 10 minutes or so, and then another set which were maintained at the dry level and watered again every 48 hours. And then finally I had another treatment which was my wet-dry cycle, where plants were dried down from the wet to the dry, subsequently re-wet. and then maintained at the wet level until harvest. So pretty complicated, but you'll understand a little bit more later. So in addition to my water treatments, I also had 3 nitrogen treatments. I used urea as my fertilizer, and this was pre-mixed into the soil before potting, and then no more nitrogen was added throughout the experiment. And so the nitrogen concentrations ranged from 25 to 150 milligrams of nitrogen per kilo of soil. In addition to that, I had my 4 reps and then 1 harvest time point, giving me 120 plants. So with this experiment, I wanted to ask a couple of questions. So firstly, how does wheat respond to variability in water supply when you also look at the different nitrogen availability? And how do these different wheat genotypes respond differently? So does Gladius respond better or worse than Kukri, for example? So I had a couple of hypotheses that I had. So firstly, I hypothesized that the frequency of watering would actually have a greater impact on plant growth than the quantity would, and that the water regimes I imposed would then affect nitrogen availability and subsequently uptake. So onto the exciting part, onto the results. So here we have a graph showing shoot and root biomass for Gladius, one of my varieties. So what we have is shoots above the x-axis, roots below the x-axis, and then N1 is my lowest end treatment, N3 is my highest end treatment, and then they're subsequently grouped according to the 5 different water treatments. So I'd just like to highlight a few of the main results from here. So essentially, we can see that with increasing nitrogen, root biomass increased, particularly under the wet water treatment, which is kind of to be expected because this is like an optimum condition for the plant. However, this is a bit different if you look at the plants subjected to the wet 48-hour treatment, where actually you have more shoots and roots under the N2 treatment, which is really interesting. However, you look at the other end of the scale and look at the drier treatments, we can see that with the dry 48-hour watering regime, there was the smallest root to shoot ratio, which could suggest that these plants were really stressed and didn't really like this 48-hour watering period and would rather prefer some consistency in their life. Unfortunately, it was not to be. But if you then look at this, look at the wet-dry plants, which also underwent a drying process, these actually fared better than the dry 48-hour plants, and also the dry treatment plants. And this is possibly because these plants had a sort of a priming effect in that they got used to being dry, but then they were then re-wet, and so that they could recover a bit better. So in addition, I also wanted to look at the genotypic response between my 2 varieties. So on the left, we have Gladius. It's the same data that I've shown on the previous slide. And then on the right, we have Kukri. Shoots on the top, roots on the bottom. So overall, Kukri was the larger, faster genotype. But what's interesting to see here is that, yes, roots increase with increasing nitrogen for Gladius under the wet treatment, but with Kukri, you actually got a higher root growth under N2. And this pattern is then reproduced for the wet 48-hour treatment, where this 48-hour watering period actually encouraged greater root growth under N2 for both varieties. Now, if we look at the, even more variability with the wet-dry cycle, you can see that for Gladius, the wet-dry plants were bigger than the dry plants for Gladius. However, with Kukri, there was no difference, which is interesting considering it's supposed to be a drought-susceptible plant, but this could be that because it's better at taking up nitrogen, that this is sort of balancing out the water stress. So in addition to biomass, I also looked at specific root length. Now, Richard already explained what that is. It just takes into account total root length and your root biomass. So essentially, if you have a higher specific root length value, you have thinner roots. So this is Gladius, and basically, overall, we can see that the dry treatments circled here had thinner roots than the wet treatments, But interestingly enough, that you can also see that specific root length decreased, so roots were thicker with the N3 treatment, my highest N treatment, specifically shown here where these arrows are. However, this trend was not seen for the dry water treatment, which could suggest that plants were looking for water and so wanted to send out roots. Not necessarily more roots, but thinner roots, and not wasting as much carbon. So again, I wanted to compare the two genotypes, but also I wanted to compare specific root length in relation to the biomass. So if we zoom in a little bit and look at the wet-dry biomass for Gladius, we can see that the root biomass is relatively the same across the different nitrogen treatments. However, what's really interesting here is that if you look at the specific root length, there's a huge difference with the nitrogen treatments. So the increase in nitrogen, you actually get a decrease in specific root length, so you have thicker roots. And then if you look at kukri down here, again at the wet-dry root biomass, again they have a similar biomass, but they have a different pattern in terms of a specific root length. So here you actually have thinner roots under the N2 treatment. So basically this just goes to show that although they may have the same biomass, both the nitrogen and the water is having an impact, or more the nitrogen is having an impact on the allocation of roots of the plants. So to conclude, we can say that water had a greater impact on plant growth than nitrogen did. We can safely say that plants prefer consistency, which does pose a little bit of a problem with the current climate change happening. Frequency of water, to answer my question, frequency of water was more detrimental to the growth of the plants than the quantity was. Interestingly, but also maybe not surprisingly, kukri was better adapted to nitrogen uptake under any of the water treatments, but this could be because it was more nitrogen use efficient than Gladius. And also Gladius and kukri had more growth, particularly under the N2 treatment, my medium N treatment, especially when looking at the wet treatments, and also, as we saw that with the specific root length, under the wet-dry treatments. So there's definitely more to investigate in terms of maybe the highest nitrogen that you can put onto plants is not necessarily going to be the most beneficial if you take into account the sort of water treatments that you have or the rainfall. So now that I am based at the University of Nottingham, I'd like to follow on a little bit more and look at the impact of soil moisture on soil structure, and then how that impacts root formation and subsequently nitrogen uptake. And I'll be using X-ray CT to hopefully produce similar images shown on the right, which are comparing two genotypes with varying nitrogen use efficiencies, comparing Australian and UK wheat varieties. And I'd like to thank the University of Adelaide for my scholarship, for my joint award, and also the University of Nottingham, University of Adelaide, and the International Plant Phenotyping Network for the travel grants to allow me to attend this conference. Thank you.